We are joined now by researcher Daniel Koboldt from Washington University, a co-moderator at the Cancer Genetics in Genomics Era session. Thanks so much for joining us.
Thanks for having me, Sloan.
The session has already happened. Please tell us about the highlights for those who were not able to attend.
Well, it was yesterday, and I think we had 4 distinguished speakers. We had Mike Dyer from St. Jude Children's Hospital, and he started out the session talking about pediatric cancer and some of the work that St. Jude has done with WashU on solid tumors. So he covered a couple of different tumor types and some of the things that we found by doing sequencing of hundreds of tumor samples, but in particular they were looking at rhabdomyosarcoma as one of these and Ewing sarcoma, and just looking for reasons that could explain why these cells form into tumors and whether or not that can be used to help guide treatment decisions. So then our second speaker changed gears a little bit. It was Li Ding from WashU, and she talked about some of our informatics approaches that we're doing in the cloud to allow for sort of high throughput computational analysis of big cancer data sets. That was the breadth part of her talk. And then she also talked a little bit about going in depth on a single tumor. So we have this leukemia sample called AML31 where we've sequenced it to very, very high coverage. And it's interesting because when we first did the analysis of this genome, we saw about 108 mutations that distinguished the tumor from the normal cells in that patient. And when we did very, very deep sequencing, we found a lot of low frequency mutations. So we missed hundreds of mutations that were only in a fraction of the tumor cells. And one of the reasons is we didn't do enough sequencing for it. So it was a useful look at what you can learn from doing deep sequencing of a tumor. And then our third speaker was Jan Koboldt from Koboldt, and he was talking about a few different things with the ICGC, which is this complementary effort to TCGA to study common tumor types. And in particular, he was focusing on this phenomenon that we've seen in some tumor types where there's a really unique pattern of chromosomes. It almost looks like a chromosome has been shattered and then stitched back together in the cell. And so you see these, the patterns of it, you see these fragments that normally are in a line on the chromosome are randomly assembled back together. And you see copy number changes too, like some fragments are copied over and over so they have high copy number, and some are lost so they have what seems like a deletion. And he noticed that, and their group found that a lot of the things that seem to be amplified are oncogenes, so they're driving tumor development and growth. So that was a pretty interesting take on just what they've done in the ICGC. Our last speaker was Josh Stewart, who talked about the pan-cancer analysis. And basically that's where they take the tumor types that we've done for the Cancer Genome Atlas and for ICGC and put them all together and look for patterns across a variety of tumor types. And it was a really great way to end the session because what they've been doing at UCSC is building maps of tumors based on expression data and other components that we can get from genomic analysis. And when they profile tumors in that way, they can sort of cluster by tissue of origin, or they can cluster by tumor subtype. And he brought it back to just how do we use this information to help a single patient, because they had a patient with a tumor that was really resistant to the treatments that they had tried. And they did some profiling and found it was similar to a rather unexpected tumor type, and as a result changed the therapy, and the patient seems to be improving. So it was a great session.
It seems like it was an incredibly informative session. What do you hope that attendees take away from the session?
Well, I think the main message is that these high throughput genomic technologies that we have now are really changing the way that we study cancer from a genomics perspective, and also that these can really be used to not just to build these profiles of common tumors and write the big papers, but also to build resources that then we can use in the clinic to help these patients.
Daniel Koboldt, a researcher with Washington University, thank you so much for joining us.
Thanks for having me.